A-level biology 9700 paper 5 variant 2 May June 2024 question number 2 and question number 2 is tumors may be described as benign or malignant. Malignant tumors can lead to greater complications for the persons with the tumors. Early identification of tumors, particularly malignant tumors, is important for effective treatment. Benign tumor cells are and malignant tumor cells can look similar when viewed using a light microscope. Some scientists wanted to develop a diagnostic test to identify tumor cells as benign or malignant. The scientists investigated whether the diameter of the cell nucleus could be used to identify the type of tumor cells as benign or malignant. The scientists used a light microscope with a calibrated eyepiece reticule to measure the diameter of the nuclei of stained tumor cells. Figure 2.1 shows a photomicrograph of stained tumor cells viewed using a light microscope. An eyepiece reticule was placed across the nucleus of one of the tumor cells, and you can see in figure 2.1, cell surface membrane has been shown. It is labeled, and nucleus is also labeled, and the cell X has been shown, and this eyepiece reticule is kept over it. The eye uh, um, and students, the eyepiece reticule is used. It is kept in the eyepiece to with the scale, which is called as eyepiece reticule. So the image magnification is about 400. So the calibration of the eyepiece reticule is one eyepiece reticule division is 320 nanometer. So use the calibration of the eyepiece reticule scale to calculate the actual diameter of the nucleus of cell X. Show your working and state your answer in micrometer. So we are moving back. So first of all, we have to count the scale, the points in scale. So it starts at 40 and then it ends. These are 10, 20, and it is ending over here. It's almost 28 eyepiece reticule units. Now we will make use of these 28 eyepiece reticule units, and we will try to find out our answer. We will calculate in this way in the given space. So 320 nanometer. 320 nanometer I1 eyepiece reticule division is equal to 320 nanometer. So first of all, we will change this 320 into micrometer. First of all, correct divisions on eyepiece diameter, that is 28 division. So 320, we will change this 320 nanometer into micrometer. So 320 will be divided with 1000. So it will be 32 micrometer. Now we have to find the actual diameter. So the actual diameter will be equal to 28 multiplied by 0.2t. So the answer will be, if we multiply it, let me calculate. So if we multiply 28 with 3, so the answer will be 8.96 micrometer. So this is the answer. For the investigation into whether the diameter of the nucleus could be used to identify the type of tumor cell, tumors of the thyroid gland and endocrine gland are used for the diagnostic test. The scientists carried out the procedure using two different stains. So step one is there. Step one is 50 people. 50 people diagnosed with benign tumor and 22 or 24 people diagnosed with malignant tumor were selected. 50 with benign tumors and 24 with malignant tumors. So a sample of tumor cells were removed from the thyroid gland of each person. The cells were stained with either papino, stain, or pap smear test. It is, and this type you can say the pap, coleosin, a hematoxylin, and eosin stain, E and E. And samples were viewed using a light microscope with the malignant, with the magnification of 400 in each sample. The diameter of 100 nuclei were measured. So pep, papillopolyostain, PAP, or hemato xylene and eosin. So these are the two different tests which were conducted on them. The each sample, the diameter of 100 nuclei were measured. So the sample size, the measurements were made by one of the scientists who did not know the type of tumor cells they were measuring. Now the question, you can see the results of the investigation by doing these uh, papinofolio pap test on H and E stain tests. You can see over there. Tables total benign cells 50 people. They have shown the mean nuclear diameter, the standard deviation, and standard error has been shown for all types of tumors. So suggest how the scientists can standardize the method of measuring the diameter of the 100 nuclei in step 5. And what is step 5? Step 5 is in each sample the diameter of 100 nuclei was measured. So to keep standardization in step 5, so the valid comparisons can be made between benign and malignant tumors. So the proper measuring. So the and it can be done by measuring. It can be done by measuring the widest part of all nuclei. Next, identify one variable that the scientists have standardized in step 1 and 2, step 6, other than details of the method of measurement of the nuclei. So you can see when they have to uh, adopt the method and they have to follow the different steps, and during this following of these steps there are many points which should be kept in mind. First of all, there should be the same magnification when they measure them. So the same magnification, as you know, they have used 400, it should be 400 for everyone. Same magnification should be used while measuring these cells. And the same number of nuclei should be. And the scientists used a t-test to compare the mean nuclear diameter of the pap stained cells and the H and C stained cells into state a null hypothesis. And the null hypothesis, if we can make this hypothesis in this way, there is no difference between the mean nuclear diameter of the abstained cells and E and E stained cells. Next question. Next question is the t-test. The t-test, the t-value the scientist calculated had a probability value greater than 0.25. So state one conclusion that can be made about the effect of using the two stains PAB and H and C on the mean nuclear diameter of the cells. So you can say state one conclusion, effect of using the two stains. So in this way, we will see that we will accept there is no significant difference. So null hypothesis is accepted between diameter of nuclei. So null hypothesis is accepted. Now the next question is state and explain one conclusion that can be made from the data in table 2.1 when comparing the results for the 5,000 benign cells and the 2,400 malignant cells. So we can state, first of all we have to state, then we will explain our idea. So the mean nuclear diameter, the first statement, it can be mean nuclear diameter of benign cells is smaller than in malignant cells. And you can also see this. After writing, I will show you. I will take towards the table. So we can explain this by using our table. So we are moving towards the table. So our statement is there is the mean diameter. You can see the mean diameter is different in both of these cells. Here you can see in the benign cells. The mean diameter in the benign cells is 7.3, 7.3, and 7.2, whereas it is in the malignant cells, it is 9. And so it means it is in malignant cells, the diameter is larger as comparison to benign cells. It is smaller in benign cells and it is larger in the malignant cells. So in this way, we can explain this, we can write this, and it is, and you can say the standard deviation as well. It is the value of the standard deviations are not overlapping. They are different in both, and standard deviation values all are also different in both of these. So we are moving towards the our answer, the statement which we have written, and then we are moving the explaining. We have the given explanation over there. Now we are going to write the explanation which we have got from the table. So you can see the values are not overlapping. There is a significant difference. As it is shown in the table. So we have written, we have explained in both ways. We are moving, suggest and explain two disadvantages of this procedure. You can say this investigation was consist of many steps. First of all, to select the people who are suffering with malignant tumors or with benign tumors. So then after selecting them and then choosing them and then taking the samples, so it will take time. So this is a long procedure. So long, this is the very main disadvantage of this procedure, that is it is time taking. It is time taking as to select the persons and counting, measuring, you can say, uh, to adopt the standard method. So it takes time. So this method is time taking. And the second disadvantage can be that this investigation can be conducted by the people who are trained to get the, you can say, the samples. So, and they know the lab work properly. They are properly trained people, lab technicians, trained people are required to conduct this investigation, which is also a time taking procedure. And you can say during the long time, the people or the persons can suffer with pain or anxiety. due to tumors. So students, it was all about the question number 2. Please like, share and subscribe my channel, and thanks for watching.